Hello everyone, welcome back to the part 6 of Hematology. In this video, I will be discussing about iron metabolism. Consider subscribing to my channel for more of medical content and health lectures. Let's begin. Iron metabolism. Normal values of iron. The total quantity of iron in the body is about 4 grams. The approximate iron distribution in body is as follows. In hemoglobin, it is 65 to 68%. In muscle, as myoglobin, it is 4%. As intracellular oxidative heme compound, it is 1%. In plasma, as transferrin, it is 0.1%. Stored in reticuloendothelial system from 25 to 30%. Dietary iron. It is available in two forms, heme and non-heme. Heme is mostly found in fish, chicken and meat, whereas non-heme in vegetable, cereals and grains. Ferric, that is Fe3 plus form, it is in ferrous form, that is absorbed. Absorption of iron. Iron is mainly absorbed from small intestine, that is mainly the duodenum. It is absorbed through intestinal cells by pinocytosis and transported to plasma. Bile is essential for absorption of iron. Now, iron mainly comes into the stomach and is mainly absorbed in the duodenum, that is the enterocytes. Iron is present in two forms, Fe3 plus, Fe2 plus. Both come to the duodenum. Fe3 plus is a ferric ion, whereas Fe2 plus is a ferrous ion. Ferric ion cannot be directly absorbed, so it has to be transformed to Fe2 plus. Now, in the lumen of the intestine, both Fe3 plus and Fe2 plus ion forms are present. Fe3 plus is transformed to Fe2 plus by DYCTB, that is duodenal cytochrome B, that is ferrireductase enzyme. This Fe2 plus is transported inside the enterocyte by DMT1, which is divalent metal transporter 1. It transports only Fe2 plus ions. It is transported along with a proton. Fe2 plus, it is very toxic in nature. So it is stored in non-toxic form, that is Fe3 plus. This Fe2 plus ion combines with the apoferritin inside the enterocyte to form a compound, that is ferritin. That is, the ferrioxidase activity takes place over here. Some of the Fe2 plus ion is transported out from the enterocyte into the bloodstream through the ferroportin. Here again Fe2 plus is transported back to Fe3 plus by the hephaestin. It acts with the apotransferrin in the blood and forms the iron apotransferrin, which is later stored in the blood or used for erythropoiesis. So basically what happens is, in the enterocyte, DYCTB transforms the Fe3 to Fe2 out in the lumen, and that is transported inside the enterocyte by DMT1 transporter, which forms the ferritin compound and later transported into the bloodstream and again transformed back to Fe3 plus by hephaestin and forms the Fe apotransferrin used in stored or erythropoiesis. Daily loss of iron. Males, 1 mg of iron is excreted every day through feces. Now, whereas in females, iron loss is high. Now let's see why. 1 gram of hemoglobin contains 3.34 mg of iron. So 100 ml has 15 mg of Hb and 50 mg of iron. So basically 100 ml of blood contains 50 mg of iron. So in females, during every menstrual cycle, 50 ml of blood is lost, that is 25 mg of iron. In hemorrhage, 225 mg of iron is lost. If you find this video helpful, let me know in the comment section.